I'm recording again, and here in the problem that they give you, where they're asking, they give you some original DNA, and they say, here's this DNA, and they're like, okay, you're going to go through some PCR, you're going to do some replication, DNA replication, and you're going to make 2 new strands. When you're doing this, though, you're going to change, and remember, this is the classic experiments of biology that we've gone over. There's a couple postings already of it. But you're going to add some different kinds of nucleotides so that instead of a blue, in this case blue, right? But instead of the original DNA, you're going to be able to tell what the new strand looks like. And so in the problem, they use yellow and blue. So that's what I'm going to use. I'm going to use yellow and blue. The yellow is the new strand, the blue is the old strand. So this is the original DNA, right? Now, when you do DNA replication, what happens is you do semi-conservative replication. We've done this when we went over the classic experiments in molecular biology. And what you get is you get that replication bubble. Those of you that remember how to do DNA replication, you get DNA polymerase coming in, right? Do you need to know all this? No. You just have to remember what semi-conservative means. But to make sure you understand it, I'm explaining it. So you're going to get a replication bubble, remembering that you have to open up the strand. The strand has to be unwound, right, so that you can actually access the code. The code is a nucleotide base that's off the sugar phosphate backbone. So you're going to have this open kind of big, what we call replication bubble. And DNA polymerase is going to be in that. And wherever you have an A, let's say I have A, T, T, G. Over here I would have had what? If A, right, it would be U if it was RNA, but this is 2 DNA strands. So where A is matched up with T, and then A with T again, and T with A, and T with A, right? And this is C, right? So those are the original strands that were used to be put together, but DNA polymerase separates them and starts to read the original. It reads this one, and then another one starts to read this one in the other direction. Is that clear? But these are going to make copies, right? So where there's an A, there's a what? There's a T, and where there's a T, and there's an A, and there's an A, and there's a C. Now this is a yellow strand. Do you see it? And it's going to continue to make that. And it's yellow because in the classic experiments in molecular biology, they use radioactive nucleotides to make the new ones. So they could tell by mass which ones were new or half, and which ones were really heavy, or which ones were really light. And they could tell which ones had 2 original strands, which ones had half and half, and which ones had 2 new strands. So in the first half of replication, what you're going to get is, obviously here you're also going to produce, and one's going to be a lagging strand, one's going to be a leading strand, but in any case, you're going to produce A, T, T, G. And so you get 2 new strands after 1 round of replication. You get 2 new strands. You have 1 original strand, right? 1 original that's blue, and the other one's going to be what? Yellow. Yellow. And why is that? Because it's semi-conservative. If you want details, watch the videos that we've posted in the past. I think I've done this like twice or 3 times this year. Look at the videos we posted on the YouTube channel, and look at the classic experiments of biology, and you'll see everything explained from beginning to end. But this is semi-conservative. And you're going to make 2 of them. Semi-conservative meaning you're keeping 1 of the 2 original strands. So both of them look like this. Now that should make sense, I think, to everyone. But now what happens, this is the first round. What happens when I do it again and I go after the second round? What's going to happen is I'm going to make 2 copies. The same thing's going to happen for both of them, so let's just do it with 1. What's going to happen then in this second round? You're still going to get semi-conservative, right? so DNA polymerase is going to read the blue. It's going to keep the blue, right? And it's going to make a what?
Yellow.
A yellow. Very good. But then the other one is already yellow, isn't it? The other yellow, this is already yellow. So it's going to make
Another yellow.
another yellow. Exactly. It's already, is this not already yellow? This is what DNA polymerase is going to use to read in order to make the copy. So here, when you're looking at this, you go to a fine tip here. If this was T, A, C, then the copy would, the green were A, T, T, and G. Then this is going to be used, this blue is going to be used to make this yellow, but this yellow is going to be used to make this yellow. So you'll have the first yellow having the T, that's the new original, right? A, C, and the other one, the new one, is going to be A, T, T, G, T, T, G. All right? So it's semi-conservative. Semi-conservative means that when you have an original strand, each of these original strands are going to be used to read. A new one is going to be made to link with it. So when you look at that problem, think about it and think about how this makes sense. In the original strand, you had blue, you should have a mixture, and then when you have it again using those new nucleotides, you'd expect two new strands to be. Now, there's still one old one and one new one. It's always one old one with one new one. It's just that both of these are yellow now because you made them with the new nucleotides. All right, that's it. Okay, so let me say a couple things then on this, because some people are still a little confused. That's fair. Again, I'd refer you to the original YouTube video, but what's confusing here is I'm leaving some things out. So this original version of the DNA was made with regular nucleotides. So you had a sugar, a ribose sugar, you had a phosphate, you had some kind of nucleotide here, right? Nitrogenous base, right? And you had a bunch of these linked together in a chain. This original, no radioactive. And that because it's not radioactive, they colored it blue. Is that clear to everybody so far? So when you see blue here, that means it's not radioactive. Yellow, then they took that DNA replication, and when they did DNA replication, they took that DNA those bacteria out of the test tube that had regular nucleotides, and they put it in a test tube. They put those bacteria in a test tube that only had radioactive nucleotides. So you still had the ribose sugar, the phosphate, the nitrogenous base, but they were radioactive. And they represented those with yellow. Is that clear? All right. So anywhere where the original DNA that wasn't radioactive is going to be done in blue. Anywhere where it's the new stuff, the stuff that's in the new test tube, it's going to be yellow. So I got this DNA. It's going to be replicated. And it's blue because it's not radioactive. Okay, now I'm going to put in a test tube, two test tubes. There's one test tube, here's another test tube. But there's only yellow stuff in here. It's only radioactive stuff. So as I put it in there, there's three possible things that could have happened, depending on what's happening with DNA replication. There were three possibilities as far as the original scientists could figure out. First, you could get conservative, which means in the end, when this replication happened, after you copied it, and you had the original DNA here, you only have the yellow now to use, then you would have, in the end, 2 yellows, 2 new strands, and the 2 old strands stuck together. That's one possibility. That is not correct. That would be known as the conservative model. We would not expect that because we know that this DNA replication is semi-conservative. There was another one that was called dispersive. In other words, instead of the strands staying together, maybe the machinery, the cellular machinery, just mixes it all up. So you get a little blue, a little yellow, a little blue. This is called dispersive. All right, so maybe that's what happened. Maybe it doesn't. Maybe a replica uses some of the old and some of the new throughout the whole thing, and it does that, and then it does it over again, and you get two nucleotides, right? They're like that. That would be dispersive. That's also not correct. So these are not correct. What is correct is that we know via the experiment that I explained earlier that when we put this original in a test tube with new nucleotides that are radioactive, what we get is one nucleotide that's not radioactive and another nucleotide that is radioactive in both the new copies. So because we conserved or kept one of the original strands, we call this semi-conservative. Semi means half. Semi-conservative. And that means half conserved. Half the DNA is the same, the other half's new. Half the same, half new. So when you look at the here's one semi-conservative, here's another. So now when we do it again, let's do it again. Remember, what's happening here? I'm reading this one and I'm making a new one. Do you see it? I'm reading this one and I'm making a new one here. I'm reading this one and I'm making a new one here. Now, I take this that already has one strand that's radioactive and one that isn't, and I'm going to do that whole process again. I keep this one, it's not radioactive, there it is. The new one's going to be radioactive, right? Now this one's going to read this one, and this one is already radioactive. The new one's going to be what? Radioactive as well. So this one, the whole thing is radioactive. But we would expect that because this one was already radioactive. The one that DNA polymerase matches to it should also be radioactive. So we'd expect this whole thing to be radioactive and this one of the strands not to be, because remember originally. Now this would happen, just in case you're confused, let's make sure you understand that the same thing would happen to this one. All right? It's not like, oh, that happens to that one but not the other one. No, the same thing happens to this one. You have 2 new strands. One radioactive, one not. It's just in the practice problem, they only have you do just this one version here, and in this example, I only did this branch because you see it's the same, right? Mm-hmm. So when you do DNA replication semi-conservative, you keep one strand the same, and it uses that one as a template. DNA polymerase uses that one as a template to make the second strand, and then they stick together. And you just keep doing that over and over and over again. So I hope that helped. Longer than I thought I wanted to do, but that'll do.